first thing I'll say is this has been really a collaborative effort between our center in Pittsburgh and the center at the University of Wisconsin-Madison, and a little bit of inspiration from Pittsburgh Institute for Neurodegenerative Diseases, which is where my lab is housed. What I really want to highlight is something that's been talked about already in posters, and even Jill mentioned about the impact of aging, or the fact that aging is probably one of the major risk factors for BPH LUTs. And there's been so much research on, what are the kind of the consequences or the features of normal and abnormal aging. And certainly many people have focused on the health of mitochondria, and how mitochondrial dysfunction during aging contributes to so many different aspects of cellular tissue and organ dysfunction. So kind of the question becomes, move forward. Oh, sorry. Why even study mitochondrial dysfunction and aging related to BPH LUTs or LUTD? What are we going to learn that's new? As I mentioned, age is one of the major risk factors for BPH LUTs, and mitochondrial dysfunction is well known to be one of the features of aging. But we kind of take an approach with, and I think many people are beginning to think about this, because what we don't really know, what we're learning more and more about, is the specific alterations of mitochondria that occur in the aging prostate or lower urinary tract in general. And the question is, how does mitochondrial dysfunction contribute to the pathology of BPH and the symptomology in LUTs? And more importantly, too, as basic scientists, if we have a mechanistic understanding of mitochondrial dysfunction in this particular part of the body, does that give us opportunities to develop what I'll call desirable therapeutics? I mean, that's kind of ultimately the goal, is to get better therapeutics that are going to be desirable for the aging man to take for this benign condition. So once again, as I mentioned, we've been inspired to kind of jump into this from some of our colleagues, or my next door lab mate, Dr. Tim Greenemeyer, who studies Parkinson's disease, where it's well known that one of the key drivers of PD is attributed to mitochondrial dysfunction, specifically dysfunction in complex 1 of the electron transport chain. And Tim and his group developed many years ago a pharmacological model to study ETC dysfunction and PD, and that's been to use the pesticide rotenone. Now, since Tim's pioneering studies using rotenone to kind of trigger PD-like symptoms or PD-like features in rodents, there have been more recent kind of genetic approaches, as I kind of highlight here, that also, once again, highlight that, at least for Parkinson's disease, disruptions of the complex 1 seem to be a major contributor to that. And you can see here in this little diagram, of the ETC, where rotenone kind of is acting. Now, the important thing I want to highlight is right now we are kind of laser focused on complex 1, but it doesn't mean that there are other components of electron transport chain that might play a role in what we're studying in terms of age effects on BPH LUTs. But for now, we're kind of, our lab and Rilke's lab are focused kind of on complex 1. Now, what you notice here in this little diagram is that what are the consequences of disrupting complex 1? And what you'll see is that there are a number of different consequences. NAD plus, NADH ratios are different. There's a drop in NAD plus levels because you're not driving NADH to NAD plus. There's a decrease in ATP. I keep doing that. And there's an increase in reactive oxygen species. Okay, now I'm focusing a little bit on this one component. What I show you here is what's known about the components of complex 1 in humans, and there's like 45 subunits, which makes it a little bit complicated, but also maybe an opportunistic way to study complex 1. And I'm focusing on this one subunit, NDUF3, and you'll see why in a few seconds. So I'm going to show you a couple of bits of pilot data that's been generated by our team, but also kind of talk about the challenges and some of the opportunities if we kind of think that mitochondrial dysfunction is a component of BPH LUTs and contributes to that. So these are studies we did, or Teresa Liu did in collaboration with us and Tim Greenemeyer's lab initially, looking at Lewis rats, which is what they use for their PD model, treated 5 days with rotenone. And one thing I should mention is that one of the features that is associated with the loss or reduction in complex 1 activity is a reduction in the amount of this particular component of complex 1, NDUFS3. And in the rat, we are very fortunate that Tim's lab was looking at the brains for their Parkinson's model and we're throwing out the carcasses. So we said, hey, give us a carcass, we'll take out the prostate. And we did that, and Teresa did some staining and basically confirmed what was seen in the PD model in the midbrain, is there's a reduction in NDUFS3 in these rotenone-treated rats. Teresa's gone on to do this in mice as well, and we see the same kind of thing. Now, importantly, in this rodent model, it seems like this pharmacological manipulation is mimicking in the prostate what they're seeing in the midbrain for PD model. But in human tissue, we're kind of curious to see whether the same thing happens. So looking at BPH and normal adjacent tissue, we see kind of the same thing, or Teresa did this analysis as well, is that there's a reduction in NDUFS3 and also another component of mitochondrial dysfunction in the sense of a regulator of mitochondrial function, PINK1, which is important in regulating mitophagy, which is basically the clearance of damaged mitochondria. Now, what have you learned from that, and what can we learn from other model systems? I'll show you this kind of complicated cartoon that is derived from a review that looks at what's going on in idiopathic pulmonary fibrosis. What are the contributors to that fibrotic state, which is a very severe fibrotic state, nowhere near the same as what type of fibrosis might be occurring in BPH. Now, never mind all of the names, but what I want to highlight here is the fact that it's well established in a lot of different groups that mitochondrial electron transport chain dysfunction, mainly complex 1, contributes to IPF. And it contributes in IPF due to a number of different factors, reduced mitophagy. You can see PINK1 is reduction. But what I really want to highlight here is that multiple cell types are involved. And mitochondrial dysfunction is thought to occur both in the fibroblasts, in the epi cells, and also in inflammatory immune cells. And all of those together, along with secreted factors, at least in this IPF condition, can trigger this fibrosis. Now, we've done a few experiments, and Teresa's done this, and we've done some in our lab looking at, once again, using rotenone as a way to kind of mimic complex or to trigger complex 1 dysfunction. And this is a summary of some RT-PCR analysis that Teresa has done using rotenone treatment of a stromal cell line, of human prostate stromal cell line. And you can see that there's a number of profibrotic genes that are upregulated, genes affecting mitochondrial dysfunction, senescence. We've looked in epithelial cells at junctional proteins, and we see, in many cases, a reduction in junctional protein expression by Western blot as a result of this rotenone treatment. So let's move on to the challenges, right? And the first challenge or gap is, is mitochondrial dysfunction in the lower urinary tract in aging males confined to the prostate? Certainly not. And Kathy Mendelson published a really comprehensive study looking at, changes in the urothelium resulting from deletion of PPAR gamma, which is a prime regulator of mitochondrial biogenesis. And so many, many different gene expression changes that relate to mitochondrial function. Lori Berter's lab at Pitt is studying age-related changes in mitochondrial function in the urothelium and seeing, loss of or disruptions in mitochondrial function in the urothelium. So there clearly is probably going to be some dysfunction of the mitochondria in the bladder that could contribute to, LUTs. Is there effects in the peripheral nervous system? Naoki Yoshimura from our group, from our team, or our O'Brien Center, has published a really nice review looking at the relationship between changes in neurodegenerative diseases like Parkinson's and lower urinary tract symptoms, and focused mainly on changes in the central nervous system that impact bladder function. But could there very well be changes in mitochondrial function in the peripheral nervous system that contributes to lower urinary tract symptoms? We don't know. It's something that I think would deserve some study. Another gap or challenge is, what are the metabolic adaptations to dysfunction mitochondria, let's say complex 1 or maybe others, that are compromised, if they are in fact compromised, in the lower urinary tract of aging males. Now, this is very complicated, but it doesn't matter. The point is that if you block complex 1, there are many ways that cells adapt to still drive ATP production and run through the ETC And most simply, you can see that one way to do that is to just pump more succinate, which can drive electrons into complex 2 and then 3, and then off you go. So cells adapt to disruptions, especially in complex 1. Some cells have the ability to do that by driving succinate and all these other pathways that might actually contribute to basically reducing ROS, increasing NAD+ levels, and making more ATP. So do these metabolic adaptations, are they different, or are they affected by aging? We don't really know. So in terms of how we might think about future adaptation or future directions that try to more deeply understand whether we can really prove that mitochondrial dysfunction is important in BPH lots, or whether we can use that as a way to develop therapeutics, is kind of highlighted here. Are there biomarkers that we can actually identify that relate specifically to mitochondrial function? There's been a few studies. There's one very interesting study that didn't distinguish biomarkers between prostate cancer and BPH, but it looked at exosomes and the presence of specific mitochondrial DNA mutations.
Mr. DeFranco, I just wanted to give a 15-second warning.
Yes, okay. So there's a couple of other things in the slide that highlight that we can use serum and blood from individuals to identify mitochondrial dysfunction that may be relevant to things like frailty. The final point I want to make in 2 seconds is that there are drugs that have been used in patients actually, and certainly in animal models, that allow one to bypass complex 1 and then generate normal mitochondrial function. So with that, I'm going to just quickly thank everyone who was involved, the Ricky group, our friends at the PIND, and look forward to hopefully having some questions at the end. Thank you.